Hello again, biology. Today we're going to be talking about stem cells. Stem cells are used in some different medical treatments, which we'll talk about over the next couple days. And while they do show a tremendous amount of promise, especially to treat conditions that we don't have any other method of treating, their use is quite controversial for reasons that we will talk about. So just very basically what a stem cell is it is a specific type of cell that can generate new and different types of cells, including new stem cells. So a stem cell, when it undergoes reproductive cycle and makes a new cell, it can either make a new stem cell or it can make other types of cells, and we'll talk about how different types of stem cells can make either just a very few number of different cells or many different types of cells. And just generally speaking, there are three types of stem cells. Um, they are embryonic, which are derived from embryos; adult stem cells, which come from adults; and then induced pluripotent cells. There is another type um, that is a type of embryonic stem cell, but you don't actually need to destroy a human embryo in order to get them, and we'll talk about those too. Another really important characteristic that stem cells have, besides where they come from, is how powerful they are. Certain cells are what are known as totipotent, meaning that if you take just one of these single cells and give it enough time in the right environment, that one cell has the ability to grow up into an entire human being. Uh, Basically, your cells, you started out as just a single totipotent cell, and cells lose their ability to be totipotent very early on in development, potentially after only two cell divisions. Basically, once there are a cluster of four cells, it's possible that once each of those divide and then become a cluster of eight cells, that those eight cells have lost their totipotency. It's very difficult to study embryos when they're that small, when they're that young, so we don't know exactly when that happens. But basically, one of the ways that we can get twins, and the way that we get identical, is a single L is fertilized by a single sperm, and then that one resulting cell divides once into two cells that are genetically identical. Each one is still totipotent, and then something happens and those two cells physically split from one another, and they each grow up into an entire human being, and each of those people has the same DNA as their identical twin. Pluripotent cells don't have the ability to become an entire person, but they can become basically any type of human cell. We do have to give them the right environment in order to do that. They won't just become anything on their own. They sort of have to be taught what to become. And like I said, they can become anything. They can become, blood or muscle or nerve tissue or skin tissue, just about any tissue in the body. But it's that one cell can't become an entire person. And then there are stem cells that are multipotent, that can grow into a few different kinds of differentiated cells, but not all of them and not many of them, and not certainly not an entire person. So this is like a stem cell that can become any type of skin cell, but can only become skin cells. Or there are stem cells in your bone marrow that can become any type of blood cell, or can create any type of blood cell, but can only create blood cells. So the stem cells in your skin can't make blood cells. The stem cells in your blood can't make skin cells, and they are multipotent. They can make a few different kinds, but not all of them. So the reason that stem cell therapies and stem cell technology is so intriguing and so worth pursuing despite its controversies, because it has an incredible potential to regenerate damaged tissue. And there are certain injuries that our bodies just simply don't recover from, particularly nerve damage and damage to heart muscle. Although there are some other kinds that we really struggle to recover from. The big two that stem cells show a lot of promise to heal and to treat are damage to nerve tissue and to the heart. So people who, let's say, break their back and have damage to their spinal cord are often paralyzed, sometimes from the waist down, sometimes from the neck down, and those injuries you simply can't recover from. Those cells don't replicate themselves in any sort of time frame that makes it so that wound can heal themselves. What we have seen using stem cells in animal trials is that we can take a mouse that has had its spinal cord severed and its rear legs are paralyzed. We can treat that mouse with stem cells, and that mouse can not heal 100% completely, but can heal enough that it's able to walk again. And certainly that's something that we would like to be able to do with people who are paralyzed. And it's the same thing with a heart attack. If someone were to have a heart attack, basically some of the muscle tissue around their heart usually ends up dying, and heart muscle, unlike other well, muscles don't heal all that well to begin with, but heart muscle in particular is very, very difficult, very, very slow to heal. So once someone has suffered a heart attack, basically their heart is permanently damaged, and stem cells again can repair that damage. Uh, there are some drawbacks to the use of stem cells besides sort of the reasons that they're controversial. Uh, Growing them in a lab is very difficult, it's very expensive, it's very labor intensive. So, talking about some of the different types of stem cells and starting with the ones that both show the most promise and hold the most controversy are embryonic stem cells. So, embryonic stem cells can become any type of specialized body cell. I can take one embryonic stem cell, turn that into a whole bunch more of embryonic stem cells, and then turn those into any type of cell that I would need. Embryonic stem cells haven't really learned to follow their own genetics yet. There are some changes that they're capable of making so that they fit whatever patient we use to treat them. Unlike a blood donation or an organ donation, where the donor and the patient need to be a close match, embryonic stem cells, you don't need a close match. I can treat any person with any embryonic stem cell. And that's hugely important because not all stem cell treatments are like that. But despite their promise and their usefulness, embryonic stem cells are highly, highly controversial because to generate new strains of them, uh, you have to destroy a fertilized embryo. There's simply no way to get these very, very powerful cells without destroying a structure that's known as a blastocyst. So, what a blastocyst uh, is a human embryo. It's about 5 days old. It's smaller than the head of a pin. It's smaller than the period at the end of a sentence. It's a cluster of about 150 cells. So approximately 5 days after fertilization, the blastocyst itself is mostly hollow. There's sort of a bubble of outer cells and then this inner cell mass. And that blastocyst, if it is, if it ends up being implanted in a woman's uterus, will become a human. So this structure is cellular. It is absolutely alive genetically. It has all of the DNA to be a person. And some people feel that basically humanity starts at fertilization, or some even before then. Others say no, it happens later in development, that these blastocysts, yes, they're alive, but because they can't survive given the right conditions, they don't deserve all of the legal protections that that humans have. And exactly where we draw that line of, okay, well, this is where our humanity begins, really plays a hugely important role in our societal decision of whether we should use embryonic stem cells or not. Because if you do believe that our not just our life but our humanity begins at fertilization, then in order to treat one person, we have to kill someone else, and that is generally speaking not acceptable. And we will continue to talk more about that. And different people feel different ways, and some people feel very strongly in one way or the other that no, we need to be protecting these blastocysts because they're human. Um, we need to be treating them as if they were babies because they are essentially, in their view, very young babies. And then other people would say, well, no, they're still just this very small cluster of cells. They haven't reached the level of complexity where they are human. And as we'll talk about, we make a lot of these blastocysts in fertility clinics, uh, so many that these clinics basically don't have room to store them and they end up getting destroyed. And other people feel that, okay, if these blastocysts that we're making are just being essentially thrown away, why not use them to treat people if we can? Especially because in some people's view, they are not human. So these blastocysts, like I just mentioned, where do they come from? If we are potentially using them for treatments, well, they have to come from a fertilized embryo. Where is that done? Well, it's done mostly in fertility clinics. So, two people who can't conceive a child naturally can go to one of these fertility clinics and um, do what is known as in vitro fertilization. In vitro means in glass. So this is essentially making test tube babies. But for every couple that does that, they typically make many more embryos than the mother in that couple could ever possibly use by herself. So these clinics end up with extra fertilized embryos, some of which they can kind of look at and see, okay, they're not viable. Um, Certain people choose to use fertility clinics because they have genetic disorders that they don't want to pass on to their children. So some of these fertilized embryos have those disorders, some of which are basically 100% deadly, lead to a very shortened life, a reduced quality of life. So a couple would choose to not use those embryos that are affected by these genetic diseases and instead choose to use healthy embryos in order to eventually impregnate the woman and then grow and have their own children. So right now, couples can choose to donate any extra embryos they have for stem cell research. And these fertility clinics are basically overrun. They have more, they have so many customers that they just have too many of these extra embryos and nowhere to store them. And basically, we end up destroying a lot of these embryos anyway. And to create storage facilities that could house them all potentially indefinitely would be very difficult to do and would make fertility treatments basically prohibitively expensive. So, if we're going to help all of these couples who can't conceive naturally have children, the end result is that some of these extra embryos will be destroyed. So, some people are just against in vitro fertilization in general because of this. There isn't really a way to do in vitro fertilization without creating extra embryos and then without those embryos ultimately being destroyed. So we do still have a little ways to go with this, and there will be a part two to this video.